After that procedure you had.
Are you okay?
I'm good. I suppose I had some viral gastritis or something. I'm not convinced.
No, you do not have to demonstrate. You do not have to demonstrate all the illnesses that we can get.
I'm trying to give you guys real life examples.
You got that old 2-bucket syndrome?
Not quite. It was bad enough.
You were like, do we think it was a prokaryote?
Yeah.
I would highly recommend not to showcase the prion diseases.
Yeah, the prion.
Yeah. Oh.
Yeah,
It needs
that's
some
a
Zofran
no-go.
stat.
That's a
It's all
no-go.
under that.
Well, they thought it was my heart at first. My blood pressure dropped to 75 over
That's what I'll
40
upload.
and had a glucose of 53.
Oh,
Wait, I missed that first part. What?
Blood pressure is 75 over 40 and glucose is 53. So I went on a little ride to Union. I was there about 8
50?
hours. They said, no, it's not your heart, but you know, you probably have a viral infection. So
Thank goodness your heart was okay.
And sugar at the same time.
I did. But I was determined to come back and see you guys because I didn't want us to get too far behind. So what we're going to do today is finish chapter 14 and they will review. The test should be next Tuesday. Keep an eye on your emails though, because my wife has to have a PET scan next Tuesday. But depending on that situation, I plan to be here, but we will see. So family's going to come up. But and this semester will get better, I promise. Okay, so where we left off, we were talking about chapter 14, the disease chapter. What do you remember of what we talked about?
Etiology, the causes of diseases.
So what's the difference between infection and actual disease?
Infection
Correct. So infection means you actually have the pathogen in your body, but disease actually is making you sick. And I think COVID-19 is probably the best example of that. So COVID-19 infected like a young healthy person who's like 20 without any medical conditions. Often they never got any symptoms. But if COVID-19 affected an elderly patient who has a compromised immune system and heart disease and COPD, unfortunately and tragically, they got very sick. So that's one key aspect of it. And then what's unique about infectious disease compared to other types of diseases?
It's only one cause.
And what is that?
the pathogen.
Yes. So most diseases have what we call multiple etiologies. That means there's multiple causes. Etiology is just a fancy word for cause. When you see that, just think of cause. But medically speaking, they're always going to use the term etiology. So the majority of diseases, heart disease, diabetes type 2, cancer, COPD, they're going to say there's a genetics component, lifestyle component, environmental exposure component. You know, if you're a coal miner, your risk of lung cancer goes up like 20%, which I would never recommend. But that's not true for infectious disease. Infectious disease is the outlier. The only reason you get sick from infectious disease is you got infected by a pathogen. And I said it, the other day, Ebola doesn't cure if you smoke. Ebola doesn't cure if you go to the gym. Ebola is going to infect you and you're going to get sick. Now, how healthy you are might determine your prognosis, because obviously in most cases a younger, healthier patient with a stronger immune system is probably going to fare better than, say, an older patient who's immunocompromised and has co-medical conditions like heart disease, diabetes, etc. But as far as actually getting infected, the only cause for infectious disease is your body got infected by a pathogen. That's it. So it is the exception to the rule. Okay, so let's talk about the nature of infectious disease. You remember Dr. Robert Koch? He was the gentleman who proved germ theory in chapter 1. He did it because he discovered the germ that caused anthrax, and he showed it to other doctors and scientists. And remember I said he had 4 postulates that you didn't have to know at that time, but you need to know later in the semester? Unfortunately, now is later in the semester. Now, you should know his 4 postulates for the upcoming test. And let me just go over these with you. First of all, postulates just means rules. That's all. Postulates is an old fancy word for rules. So these are Dr. Koch's 4 rules, how you would prove a particular pathogen caused a particular disease. He came out with these, I think, in 1888. First, let's say you take some animals, and 3 of them are sick, 1 of them's healthy. Well, in the animals that are sick, you should be able to culture the pathogen you suspect and grow it in a petri dish. And the animal that is healthy, you would not be able to culture that pathogen from their blood. Next, you would take the pathogen from the sick animals and grow it in a petri dish. Step 3, you would take the pathogen from the petri dish and you actually would inject it into the healthy animal. And step 4, that once healthy animal should now get sick, and you should be able to culture the same pathogen from the blood of the once healthy, now sick animal. If you could meet all 4 of these postulates, to Dr. Koch, this was conclusive evidence that this pathogen caused this disease. Now, a couple comments I have to make in fairness to Dr. Koch. For 1888, this is brilliant. For 140 years ago, when people were still stoning people for witchcraft and stuff, this is brilliant. This is like Stephen Hawking's brilliant for 140 years ago. Very objective, very scientific analysis, scientific method. But does this hold up in 2026 now? It does not hold up in 2026 for a variety of reasons. One, some pathogens can't be cultured in the laboratory. Prions are one. Good luck ever trying to culture a prion. Also, viruses. Can you grow viruses in Petri dish? No, you need cells like eggs. Also, some infectious diseases are caused by multiple pathogens. Who's heard of sepsis? What is sepsis?
Infection that can be everywhere. Anyway.
Used to call it blood poisoning. I'll be honest, when I was in med school 20 years ago or so, they still called it blood poisoning. Sepsis is basically when you get usually multiple pathogens in the bloodstream and they go throughout the body. It's bad. Because somebody's septic, they're in the ICU getting IV antibiotics out the wazoo. So if you were to take a blood culture of somebody who's septic, I guarantee you grow at least 3 different pathogens, if not more. Probably E. coli, staph, maybe a fungus like Candida albicans. So you can't so certain infectious diseases are multiple pathogens, not one. The other problem with Koch's postulates is ethical considerations. Can I just go injecting healthy patients to see if they get sick and die?
Sure, yeah.
Well, the medical board kind of frowns upon that. Interesting, though, when I was at Tulane Med School, they did that in the 30s. It's very illegal. But patients that were committed, they would take blood of schizophrenics and inject it into non-schizophrenic patients to see if they would start hallucinating. And the fact is, a lot of them did. So this led to research in the 90s that I had to do as a research project. We weren't injecting people, But the theory was some types of schizophrenia were caused by a retrovirus similar to HIV, but it affected the brain, specifically the dopamine neurons. It pushed too much dopamine out, which is what causes schizophrenia. So it was really bizarre because they could take blood from some types of schizophrenics and they'd find positive protein markers for retrovirus. So unethical, yes, could never do legally today, but 100 years ago they pretty much did what they wanted. Honestly, and I hate to say it, especially if somebody was committed, their rights are pretty much gone. But yeah, one of the famous psychiatrists at Tulane in the 30s would take blood from schizophrenics, inject it to somebody who was depressed, and the depressed person's hallucinating. They're like, oh yeah, all right. But obviously that's a big no-no today. You could not do that. So, true story. So the point is, Koch's postulates I do think are important because this is kind of the beginning of the scientific method in medicine. This was a very well thought out, objective, let's see if this suspected pathogen really causes this disease. So once again, for 1888, about almost 140 years ago, this was brilliant. This was absolutely brilliant. Remember, people are still blaming disease on witchcraft and bad air and swamp gas and everything under the sun. So I give him credit. This was brilliant for 140 years ago and kind of set the stage for the scientific method in modern medicine. That's why I think it's important. But it doesn't hold up in 2026. There's too many other factors. Does that make sense? Okay.
They still do that in rats, right?
You can in certain experiments they do, but you can't really extrapolate it to people just for a variety of factors, mostly ethical considerations. Now, there's some terminology when describing infectious agents or pathogens. Pathogenicity and virulence. Pathogenicity means the ability of the microorganism to cause the disease. So this is the probability that if you get infected with this pathogen, you're going to get sick. But then there's virulence, which is how sick do you get, basically. What's the degree of pathogenicity? Usually if something's more virulent, it means you're going to get sicker. And there's a lot of reasons for that. I'm going to talk about that. So, for an example, some of these pathogens may be pathogenic and they're easy to catch, but they may not make you very sick. Whereas you look at Yersinia pestis, which is the bubonic plague, that's extremely virulent. So not only is that easy to catch, if you catch it, there's a good chance it can be fatal if not treated. Now nowadays, we do have antibiotics that would treat that, but obviously in the Middle Ages they did not. And that's why it was so devastating. What could make a pathogen more virulent? The book makes this way too complicated, and it's really easy. So I'm going to give you guys the easy version, because the book, in my opinion, makes this just way more complicated than it needs to be. Pay attention to what I write, because this is going to be on your test and your final exam. I'll just go ahead and tell you. Gram-positive bacteria. What's that mean?
Thick cell
Thick
wall.
cell wall. And that would be some examples would be staph or strep. What makes them more virulent, meaning more likely to make you sick, is they release exotoxins. All right, you're probably wondering what's an exotoxin. An exotoxin is basically a digestive enzyme. It's a digestive enzyme that tries to break down proteins. because the bacteria likes amino acids, basically. And that's why staph and strep have a good chance of making you sick, is because of these exotoxins. For example, Staph aureus releases an exotoxin, and this is what can give you food poisoning. Remember the example I gave before, if you were 4th of July, family picnic, and somebody's potato salad with mayonnaise out in the sun for 2 hours, would you eat it?
No.
Hopefully not. Because chances are it's been infected by staph. Staph will try to break down all the proteins in that mayonnaise and eggs to absorb those amino acids. So it's going to release an exotoxin, which is a digestive enzyme. If you ingest that exotoxin, you're going to get horrible food poisoning. I'll leave it at that. So that's one example. Strep pyrogenes is the pathogen that gives you strep throat. That's bad enough, but if it gets in your tissue, like let's say you have a cut, you get strep pyrogenes in your flesh, it releases that digestive exotoxin that can give you necrotizing fasciitis. What's that? That's flesh-eating bacteria. Strep pyrogenes is one of the most notorious bacteria for flesh-eating bacteria. It's not the only one. There's a few others. But it's one of the main ones. Because bacteria love two things. They love glucose. They love amino acids. So if strep pyrogenes gets into the actual deep tissue, it's going to start releasing an exotoxin, which one, again, an exotoxin is just a digestive enzyme to try to break down all the proteins it can, and then it tries to suck up the amino acids like a milkshake, basically. But that's what causes necrotizing fasciitis if somebody gets strep pyrogenes in a wound. So this is why some gram-positive bacteria are more virulent, are likely to make you sick, because of the possibility of exotoxins. These are exotoxins they're releasing into the environment so they can break down cells and proteins and get amino acids, all that. Now, gram-negative bacteria, they have what are called endotoxins, and these are a little different. So first of all, what's a gram-negative bacteria? It's a thin cell wall. And probably the most famous is E. coli. What happens with some gram-negatives is, if you remember, a gram-negative has a really thin cell wall, but then it's got a little outer membrane on top of that. And on top of that outer membrane are these molecules called glycolipids. They're like half sugar, half fat. And in some cases, if this bacteria were to die in your body, those glycolipids get released into your bloodstream and cause an anaphylactic reaction. What's that? A very severe allergic reaction. And I think typhoid is the main culprit here. So with an endotoxin, not every gram-negative has an endotoxin, but the ones they do, these glycolipids, or sometimes they call these lipopolysaccharides. They mean the same thing. They're half sugar, half fat. If this bacteria dies, it releases that glycolipid, and then your body potentially could have a severe allergic reaction to that, including anaphylactic shock. So those gram-negatives would be more virulent or more likely to make you sick. Does that make sense? Okay. So know the difference between exotoxins with the gram-positive bacteria and endotoxins, the gram-negative bacteria. And those are two of the big factors that determine if a pathogen is more virulent or more likely to make you sick. Does that make sense? All right, everybody with me?
Yep.
Okay, cool. Okay. Now let's talk about the stages of infectious disease. There's 5 stages of infectious disease you'll need to know. I'm going to go over these, but they're the incubation period, the prodromal period, illness, decline, and convalescence. And this is basically it, this chart. Incubation period means the patient's infected with a pathogen, but they don't have any signs or symptoms. So this is where a patient is asymptomatic. And depending on the pathogen, this may last, 2 or 3 days. It may last a couple weeks, may last a few years. Like how long can someone have HIV before they start getting sick? 8 years. So that's kind of the exception to the rule. That's why AIDS unfortunately spreads so rapidly, is patients could have the virus for 8 years before they start getting sick, and by then they've had multiple partners, et cetera, et cetera. After the incubation period, you get what's known as the prodromal period. And this is where the patients give you very vague generic symptoms. And I admit, as a provider, this stage is a little frustrating because the symptoms are so generic. It could be 300 different diseases. The patient's always going to say, I feel tired, I feel run down, I don't feel like myself, I've got a mild headache, stuff like that. And trying to be empathetic, it's like, okay, that narrowed it down to about 450 things it could be. And you're trying to get more specific information, there's just not info there. So they're starting to get sick, but unfortunately, the symptoms are so generic, it's really hard to make a diagnosis. I guarantee you fatigue will be one of them, pretty much. 100 times out of 100. Now, illness is the spike. This is where they're getting the full-blown signs and symptoms of the disease. By the way, you guys tell me, what's the difference between symptoms and signs?
Subjective.
Correct. So they're just saying symptoms are the subjective complaints of the patient. I've had a headache for 3 days, my stomach's been upset for 3 days, I've had chills, whatever they tell you, those are symptoms. Signs are what you can measure and observe as a provider. So do they have a fever? Do they have an elevated temperature? Is their blood pressure up? Is their heart rate up? Do they have a rash on their skin? Do they have an open wound, etc.? Are they having nausea and vomiting? So the illness is the peak. This is where they're sickest of the sick. Decline is when they're starting to improve. They're still having some signs and symptoms, but they're not as bad. They're getting a little better. And then convalescence is really recovery. They basically have passed through the disease stage and no longer have signs or symptoms. I think convalescence honestly should be called remission, personally. But, tomato, tomato. All right, so those are the 5 stages of infectious disease. So, any questions on that? Okay. One thing I want to mention, the longer the incubation period, usually worse for the population as a whole. What do I mean by that? Yeah, it's more time to spread. So that kind of goes back, syphilis can take 3 weeks to become symptomatic. And that's unfortunate. Somebody could pass that on to multiple partners. HIV can take up to 8 years. So once again, with the AIDS epidemic, that's why so many people got sick, is folks were infected with HIV for 8 years and didn't realize it. So the longer the incubation period of the disease, usually the worse it is for the population as a whole, because honestly, a lot more people are likely to get infected. Okay. Now, how do pathogens leave the body? Well, pretty much the same way they enter. They can leave through body secretions, the mucous membrane, nasal secretions, saliva, tears, blood from broken skin, feces, urine, etc. So they can basically exit the same way they enter. Now, how do pathogens get transmitted? And we've already talked about the different reservoirs. You can have animal reservoirs for pathogens. You can have human reservoirs for pathogens. You can have environmental reservoirs for pathogens. Now we're going to talk about transmission. That's how a pathogen goes from one person to the other. And there's 3 types. There's contact transmission, there's vehicle transmission, and there's vector transmission. Vector is basically insect transmission, but I'll explain that in a moment. So what's contact transmission? Well, pretty much what it says. You either come into direct contact with somebody who's infected. You have indirect contact transmission, usually pathogens spread by a fomite. Who knows what a fomite is? All right, write this down. A fomite is an inanimate object that can potentially spread a pathogen. Would you use somebody else's toothbrush? No, that'd be gross, but that would be a fomite, because that could pass germs. If somebody's sick, should you drink out of their drinking glass? No, that drinking glass is a fomite. So those are examples of fomites. Any inanimate object that can spread a pathogen is a fomite. And then there's droplet transmission. What's that?
Sneeze.
Yeah, that's when the little kid sneezes in the elevator on you and doesn't cover their mouth. And because you're a doctor, you try not to beat them up because you're pissed off. But yeah, droplet transmission is basically spread of pathogens and droplets of mucus by coughing, sneezing, if somebody does not cover their mouth. How far can droplets spread?
A long way.
3 feet.
Yeah.
So 3 feet, yeah. That's why when COVID was rampant, how far away are we supposed to be from each other?
6 feet.
6 feet. Is that a random number they just picked out? No. They knew the maximum spread of droplets is 3 feet, so they doubled it. That's all they did. So if somebody sneezes in her mouth, the germs go about 3 feet. We're going to double that. Make sure you're at least 6 feet away. So that's how they came up with that number. It makes sense. One of the few things they got right. One of the very few things they got right. I said that. Enough. Vehicle transmission. Once again, I'd say vehicle transmission is really environmental transmission. Some pathogens can be airborne, meaning they can basically float in little droplets of water in the air. The best one I can think of is Legionella. Remember Legionella? That's the one you find in standing water and poorly maintained air conditioning systems. That's how it spreads through little droplets in the air. And it can go beyond 3 feet. That's the deal. Airborne transmission means it can go beyond 3 feet because it's actually being transmitted in droplets of water in the air. Waterborne transmission means basically what it says, contaminated water. So water that's got cholera in it or Giardia or some type of pathogen. And then foodborne transmission. If food is not properly cooked or prepared or refrigerated, it can become contaminated. It can also become contaminated if the person goes to the bathroom and doesn't wash their hands, not to be gross, but factually speaking, that's a very common cause of food poisoning. And then bodily fluid transmission. You come in contact with bodily fluids, blood, urine, saliva, those can all potentially carry pathogens. So that's why good sterile technique and wearing those little hazmat suits is always important, and gloves. All right, so that's vehicle transmission. I just say that's environmental transmission, food, water, air, etc. Vector transmission is really insect or arachnid transmission. We've talked about that, the mosquitoes, the ticks, the fleas. Those are the biological vectors. They transmit pathogens from one host to the other. And here's an easy way to remember. Biological vectors are insects or arachnids that actually bite you. So think biological, bee, bite bee. So these would be your mosquitoes, your ticks, and your fleas. By comparison, mechanical vectors don't bite you, but they drag their dirty little legs through your food if you're not careful. Those would be like flies and roaches, would be mechanical vectors. They don't bite you, but they usually kind of hang out in filth, so they're covered in germs, and that's why you don't want them around your food. So with biological vectors, which one's the worst offender?
Mosquito.
Mosquitoes. And like I said the other day, here's a whole list of diseases they can transmit. Malaria, yellow fever, dengue fever, viral encephalitis, that would include West Nile virus, Zika virus, and about 40 others. So mosquitoes are definitely the worst offenders. They take a pathogen from one host, like an animal, and then they bite you and transmit it to a person. This is why you do not want like standing water or old water around your house, especially in the summer. That's pretty much just a mosquito breeding ground, so you don't want that. Probably the second culprit is ticks. Ticks can spread Lyme disease, they can spread Rocky Mountain spot fever, and there's a few others. Coming in number 3 would be fleas. Fleas are very good at spreading the bubonic plague, unfortunately. They can also spread typhus, which is typhoid disease. Okay, so those are the worst biological vectors. Mechanical vectors, once again, these are insects that don't bite you per se, but they tend to have lots of germs or pathogens on their legs. And that would be houseflies and roaches. They're notorious for carrying Salmonella, E. coli, and Shigella. So you don't want them crawling on your sandwich, basically. So that would explain transmission. Okay. Let's see what else. We're talking a little bit about epidemiology. Epidemiology is the study of where and when diseases occur. And there's some different definitions here. An endemic disease is one that's expected. The cold or the flu. Every winter, so many patients get sick with the common cold or the flu. That's nothing the CDC really gets worked up about. So that's an endemic disease. That's a common disease. A sporadic disease is an unusual disease, but it doesn't affect many people. Just could be you get 1 or 2 cases of typhoid fever or something. It's like, okay, that's a little unexpected, but you only have 1 or 2 patients that are sick, and it hasn't been widespread. So that would be a sporadic disease. An epidemic's a little different. An epidemic is an unexpected disease or a new disease that's affecting many, many people. from hundreds to thousands. And that's what COVID-19 was. But then it became a pandemic. What's a pandemic? It spreads to more than one continent. So basically, an epidemic is you have many cases of a new or uncommon disease. A pandemic is that new or uncommon disease has unfortunately spread to a second continent. So think of COVID-19, originally started in Asia and then jumped to Europe and then to the United States, then pretty much all over the world. So that's the difference between an epidemic and a pandemic. And remember John Snow? Yeah, he was the doctor who solved the cholera epidemic. This is kind of a reputation. Dr. John Snow, 1850s, horrible cholera epidemic. Hundreds of people were dying. So he took a map of London and he put a dot on every house where somebody had died from cholera. And he noticed the vast majority of cases came from this one area on Broad Street where they all got their water from the same water pump. So he went down and destroyed the water pump and sampled the water, and it was filled with cholera bacteria, basically, Vibrio cholerae. And by doing that, he probably saved a few hundred lives because folks are no longer drinking that contaminated water. So just the way he did this, kind of the scientific analysis, like, okay, let's see where are folks dying, what's a common source of transmission? Oh, they all get their water from the same water pump. I mean, I know today would be kind of like, well, duh. But 1854, that was actually pretty darn smart. So that's why Dr. John Snow is considered the father of epidemiology. Without him, there'd be no CDC or the WHO organization or anything like that. Okay. Last but not least, we're going to talk about nosocomial infections. Nosocomial infections used to be called healthcare-associated infections. Now they've given it the fancy name nosocomial infections. So what is a nosocomial infection? It's an infection somebody gets while in the hospital or a nursing home or another healthcare setting. And if you think about it, it's actually a miracle we don't get more healthcare associated infections. Why do I say that? Well, yeah, think about a hospital setting. You got lots of sick patients. Many are immunocompromised. There might be kids, there might be older folks, there might be cancer patients on chemotherapy, or lupus patients on chemotherapy. So you got a lot of immunocompromised patients. Then you got a lot of germs. No matter how much you try to sterilize and sanitize things, there's always going to be lots of microorganisms in the hospital. Then you add in the frequent interactions between staff and patients and patients and patients. And honestly, it's a miracle, quite frankly, that we don't have more healthcare associated infections than what we already do. Because a hospital really is a perfect storm, if you will. So that's why these are important to try to avoid. And what is the most efficient way to avoid transmitting a healthcare associated infection? Hand washing. I tell you this now, when you get to nursing school, because I have taught in the nursing school a couple semesters, they're going to beat you over the head with this. The number one way to stop transmitting pathogens to patients is hand washing, hand washing, hand washing, hand washing, hand washing. That's always the answer. You can wear gloves, that's great. You can use the little gel thingies, that's great. But hand washing is really the most effective way. And I'm not joking when I say they will beat you over the head with that. When I was in med school, you had to wash your hands before you saw the patient and immediately after you saw the patient. If a attending physician saw you didn't do that, they failed you for the semester. That was like automatic F. That's how serious they were. They're kind of jerks too. But seriously, the point stands that that's the best way to prevent spreading germs and giving someone a healthcare associated infection. So just remember, hand washing is number one. Okay. And with that, that is the end of chapter 14. So that's a little bit of a longer chapter, but I think it's a little more interesting chapter too. Any questions? No? Okay, we got a little time left in lecture. I'm going to start reviewing for your test. Does anyone not have a study guide? If you're missing a study guide, print it extra. So come see me, don't be shy. If you left it at home, you're better. If you're missing a handout, I may or may not have some prior handouts. I got plenty of the chapter 14 handouts.
You have a 14 handout? Is that what you missed?
Thank you.
Is that what
I've got extra handouts for chapters 11, 12, and 13.
Okay, so you did 13.
13? I was going to say, a bunch of people look like they're missing 13. So I always print enough for the class. There you go. The viruses and all these stuff. Anyone else need this? Okay. Okay. So what I'm going to do, and what we're going to do, we're going to start this, and we've got like another, 20 minutes or so. Then we're going to take a break, and we're actually just going to come back here for lab. I'm going to review the rest of the stuff. All right? I got one vote of support here. I'm not going to be able to go over every detail again. That would just be impossible, but I'm going to hit the highlights. How's that? The big points. Question?
Are we still doing quarantine next Monday?
Probably. I'll send an email out of that. No, wait. I'm sorry. Probably not. We have to review.
Okay.
My brain's a little scattered. No, that's probably going to be moved up to, I got to smile. Okay. That's going to be moved probably to Wednesday. I'll email something about that. So, this is what we would have done yesterday. Okay, so we're just once again going to hit the highlights here. There's not enough time to go through every slide, but we're just going to hit the meat and the potatoes here. That I think will help you. If you follow your study guide, you should be in good shape. So, chapter 11 is all about prokaryotes. And once again, you guys tell me, what are prokaryotes? They're either bacteria or archaea. Those are the only two types of prokaryotes. Bacteria or archaea. They're single-celled organisms. They're very small. And they're found everywhere. And these are some typical shapes. Most, I'll say prokaryotes are either cocci-shaped, meaning they're spear-shaped, or they're bacillus-shaped, which is rock-shaped. So I'm going to say this is like 80-85% of prokaryotes are either cocci-shaped or bacillus-shaped, spear or rod. There are some exceptions to that rule, though. Yep, you got the spirochetes, which look like a worm, because their flagella is embedded in the cell wall. So that'd be like the pathogen that causes syphilis or the pathogen that causes Lyme disease. Those are both spirochetes. And then Vibrio is another one I think is important. Vibrio kind of looks like the Amazon smiley face. It's like a curved rod. But Vibrio cholerae is the pathogen that causes cholera, which is important to know. And there's a few other ones that are Vibrio shaped. There's one that causes necrotizing fasciitis as well. I think it's Vibrio fuchsian or something. So, important to know those shapes. We've talked about endospores quite a bit. You guys tell me, what are endospores?
Escape pod.
Escape pods for DNA. What two families produce endospores?
Bacillus and Clostridium.
Yeah, Clostridium and Bacillus. Remember, this is where bacteria finds itself in a hostile environment. It makes a copy of its DNA. It wraps up that copy of its DNA with multiple layers of the plasma membrane and kind of injects it out into the environment. The original bacteria will die, but then the endospore could live for decades. And when the environment becomes more favorable, then it cracks open like an egg and gives birth to new baby bacteria. And the only two families that do this are both gram-positive. That'd be the Bacillus family and the Clostridium family. How do prokaryotes reproduce?
Asexual.
Most by binary fission. So there's three main methods of reproduction with prokaryotes. Binary fission is by far and away the most common. Snapping divisions, another possibility, or budding. So what is binary fission? Binary fission, honestly, is just a really simplified mitosis. Because there's no nucleus to worry about, it goes a lot faster. So basically, the bacteria cell makes a copy of its DNA, it stretches its cell wall, then the cell wall starts to invaginate. Eventually you get what's called a cross wall, where they're kind of completely separate. And then finally the two daughter cells break apart. So that's binary fission. How long does this take?
30 minutes.
Yeah, only 30 minutes or less, so very quickly. And that's why you can form colonies of several thousand bacteria within a couple days inside an incubator. So very quickly. Snapping division is just really a variation of binary fission. It's basically the two daughter cells are not the same size, and they tend to hang on to each other a little longer, and then eventually they snap apart, hence the name snapping division. What's budding? It's kind of unique. You see this in yeast, mostly. Yeast is a fungus. But a small percentage of bacteria will do this. They'll basically make an extension of their cell wall, which they'll call a bud, and they make a copy of their DNA, and they just kind of shove that DNA copy into the bud and then basically let it go as a daughter cell. Now, what's unique with budding compared to binary fission or snapping division?
The mother cell stays.
Yes, the mother cell stays, and you only have one daughter cell. And notice how the daughter cell is initially much smaller than the mother cell. So those are some reasons budding is very unique. The mother cell stays around, there's only one daughter cell, and the daughter cell is much smaller than the original mother cell. Okay. Now, why do some bacteria arrange themselves in chains and others arrange themselves in clusters?
Planes of division.
Basically, the planes of division of binary fission. Remember, most bacteria, I'm going to go ahead and say 85% of bacteria probably, reproduce by binary fission. And if they all divide on the same plane of division, let's say they all divide vertically, then they tend to form chains. One after the other, and Streptococcus is one of the big ones. Strep pyrogenes that gives you strep throat. If you look at it under a gram stain, you're not just going to see gram-positive cocci, but they're going to be in chain formation. Versus staph forms clusters. And we've looked at staph many times in the lab. Why does it form clusters? Because each individual cell can go through binary fission with a different plane of division. meaning some of them may divide vertically, some of them may divide horizontally, some of them might divide diagonally. So since each cell can divide through a different plane of division, they tend to cluster in a bunch, like a bunch of eggs, instead of chains. And the reason this is important is diagnostically. If you're looking at a patient's blood smear and you see gram-positive cocci in chains, that lets you know they're infected with strep and not with staph, where you would see gram-positive cocci in clusters. So this is important to know for diagnosis, if you will. Okay. When they did DNA testing, 3 major domains of life forms: Archaea, bacteria, eukarya. This is still considered relevant today, even though I'm not 100% on board, because they left out the viruses, they left out the prions. But as far as things made up of cells, yes, this is correct. The 3 domains are bacteria, Archaea, and eukarya. What was significant about this RNA testing in the 50s is, one, they distinguished that Archaea and bacteria really are 2 different families. Up until then, they just thought Archaea was weird bacteria. Oh, this bacteria grows on a volcano. It wasn't until they did the DNA testing, like, no, this is really a different species in a way. It's got a different type of cell wall. It's got different types of, it's got a different type of nucleoid. It's kind of like the difference between a dog and a wolf, if you will. So that's where Archaea became a separate life form, if you will. They also divided up the gram-positive bacterias into low GNC and high GNC. What's that mean?
Amount of guanine inside them.
Yeah, that means the nitrogen bases guanine and cytosine. So they found out that with the gram-positive bacteria, that's the bacteria that's got the really thick cell wall. Some of them had a very low percentage of guanine cytosine in their DNA, less than 50%. So those are grouped into the low GNC gram-positives. Others had a really high percentage of guanine and cytosine nitrogen bases in their DNA. So those became part of the high GNC gram-positive bacteria. So those are really what came out of this, why this was important. Archaea, once again, this is the other prokaryote. Its cell wall's not made of peptoglycan like most bacteria. It is made of a sugar protein, but it's got a different formation. Once again, these are the extremophiles found in extreme environments. Their DNA is a little different. It's actually more similar to eukaryotes than bacteria. They were reproduced by binary fission, budding. As of now, none of them are known to be pathogenic. So that's a big distinction. As of 2026, no Archaea is known to be pathogenic. And hopefully we never find one that is, because we'd honestly probably have no immunity to it. All right, we've already talked about the extreme files. They love the extreme environments, all that good stuff. Now we're going to go into specifics. Some examples of low GNC gram-positive bacteria. Remember, gram-positive means it has a really thick cell wall of peptoglycan. Would stain what color on a gram stain?
Purple.
Purple. And low GNC means that half its nucleotides do not have guanine or cytosine. So that's what's meant by low GNC. One of them is Clostridia. This is gram-positive rods that also form endospores. Clostridium family includes the bacteria that give you tetanus, gangrene, C. diff, and botulism. So all 4 of those are from the Clostridium family. The Bacillus family is another low GNC. These are gram-positive rods, very common in the soil, and they also form endospores. Bacillus anthracis is probably the most famous. That causes anthrax. And the other one they don't have up here, but I've mentioned before, is important, is what? Bacillus
Serious.
cereus. Yeah, don't forget about Bacillus cereus. This is thought to cause food poisoning and basically reheated fried rice. I don't know if I'm a firm believer of that, because I've ate fried rice reheated probably 1,000 times. But apparently there are cases in the literature where patients got sick from Bacillus cereus food poisoning. So that's the other one to know. What does Listeria do? It's another low GNC gram-positive. No, this one does food poisoning. Mostly meat and dairy products. And it can reproduce even under refrigeration. This is the one where I had the gentleman who had the Listeria meningitis in medical school. And remember how he got it? Sushi at a gas station. Yep, he stopped at a gas station in Louisiana and got him some sushi, which I think is a really bad idea. Because if you don't keep that just right, you can definitely get pathogens, and Listeria is not fun to have, put it that way. Some other low GNC gram-positive, we Streptococcus, which causes Streptococcus pyogenes, causes strep throat and scarlet fever and potentially flesh-eating bacteria. Staphylococcus can be Staph aureus, which can cause staph infections and potentially food poisoning because it produces endotoxins. So those are your low GNC bacteria I'd be familiar with. High GNC gram-positive bacteria, once again, these are bacteria whose DNA is over 50% guanine and cytosine in their nitrogen bases. Corneobacterium is important because it used to cause diphtheria. Now, many of you probably are not familiar with diphtheria because we vaccinated for it. But the early 1900s, diphtheria was a really nasty type of pneumonia. Pneumonia means lung infection. It used to kill lots of people. And they get these big pustules on the back of their throat and they couldn't breathe. It was really bad. But fortunately, we vaccinated for it for about, well, we're still vaccinating for it for about 80 years. And we pretty much eradicated it. That's Corneobacterium. Mycobacterium, once again, I'll do my little soapbox speech. This should not be listed under gram-positive. Why do I say that?
Because it's glycolic acid.
Yeah, their cell walls are made of mycolic acid, not peptoglycan. This drives me nuts every semester. They really should be in their own separate category. So think of Mycobacterium. They have thick cell walls, but their cell walls are not made of peptoglycan. Their cell walls are made of mycolic acid, which is a lipid. So this would be like Mycobacterium tuberculi, which gives you tuberculosis, or Mycobacterium leprae, which gives you leprosy. And how can you basically stain a mycobacterium?
Acid
You gotta use an acid-fast stain. That's why I don't think they should be listed with these other guys. That's just me. Okay. Gram-negative bacteria. Another name for that is proteobacteria. Proteobacteria, gram-negative bacteria, same thing. And gram-negative bacteria are what? They have very thin cell walls, and they stain. What color out of gram stain? Pink. E. coli is probably the most famous, but there's several more. So the ones I think you should know. Rickettsia, this is what gives you Rocky Mountain spotted fever. It's a gram-negative bacteria you can find in saliva of the hard body ticks. And if it bites you and gives you this rickettsia, you can get Rocky Mountain spotted fever. Neisseria is a gram-negative bacteria. There's two types. There's Neisseria meningitis, which gives you meningitis, the inflammation of the meninges, which can be fatal. There's also Neisseria gonorrhoeae, which gives you gonorrhea, which is a STI. So two different strains that cause two different diseases. Bordetella is the gram-negative that causes pertussis, which is also known as whooping cough, a very severe type of pneumonia where the patient literally sounds like a barking seal when they cough. But fortunately, we vaccinate against this with the DTP vaccine. I've seen one case of it, but we don't see as many cases of it because of vaccination. Okay. Legionella, we talked about. Legionella is a gram-negative that likes to live in stagnant water, and unfortunately can become airborne quite easily. And this is the one that causes Legionnaire's disease, which is a very serious type of pneumonia that's often fatal. And I hate to say it, it usually affects the elderly, because usually patients in a nursing home, they haven't maintained their air conditioning system in like 5 years, so it's filled with Legionella bacteria. That gets carried out in little water droplets, and then half the nursing home gets Legionnaire's pneumonia, which is bad. When I lived in Arizona, once a year, it was like clockwork. There'd be at least 2 nursing homes that would have Legionnaire's outbreak, so it's not good. What's Enterobacter mean? Entero means of the
Gut.
colon or gut. So remember, the Enterobacter is kind of like a subfamily of gram-negatives. These are the gram-negative rods you would find in the gut. Just think entero means colon. The most famous and most common is Escherichia coli. Is the full name. Much more common, known as E. coli. So that's probably the most famous of the bunch. But along with that are Enterobacter, Salmonella, Proteus, Shigella, Yersinia pestis, Klebsiella. And Yersinia pestis you only see in the guts of rodents, like rats and squirrels. But in humans, you'd see all the rest of these. Now, there's kind of good news and bad news. The good news is when these gram-negative rods are in your colon or your intestines, they're doing what? They're helping you. They're your probiotics, if you will. They're helping you digest your food and make vitamin B and vitamin K, so it's beneficial. The problem is if they get outside your gut and go to other parts of the body they shouldn't be, then they can make you sick. What happens if E. coli gets in your stomach instead of your intestines? You'll get gastroenteritis. You'll get the 2-bucket disease. Not fun. What happens if E. coli gets in your bladder?
UTI.
Yeah, you're a UTI. Or if it gets in your blood, you'll get sepsis. Same thing with salmonella. Salmonella is in your intestines, and it's helping you break down food. But if it gets in your stomach because you ate raw chicken by accident, you're going to get severe gastroenteritis. So that's kind of the big thing with these guys. As long as they stay in your intestines, they're helping you. But if they get anywhere else in your body, they potentially can make you sick. Now, Yersinia pestis is only found in the guts of rodents, but that gets in your body, it's going to give you bubonic plague, which is bad. Vibrio. Remember, what's Vibrio mean?
Curved rod.
It's a curved rod. Think the Amazon smiling face. Vibrio cholerae is probably the most famous because it gives you cholera. Vibrio cholerae is a bacteria. It's a gram-negative. It loves dirty water, basically. So you're going to find it in unclean water. And if patients drink from that, they're likely to get cholera, which is severe dysentery. And it's kind of tragic, but still thousands of people die from cholera every year in third world countries because they don't have access to clean drinking water, unfortunately. Some other gram-negatives you should know. Helicobacter pylori. Anyone know what that is?
Laura.
Oh, I'm sorry. Helicobacter pylori is also known as H. pylori. It's basically the bacteria that causes ulcers. They like to live in the stomach because it's got the right amount of oxygen in it. So if Helicobacter pylori gets in your stomach, it eats away at the mucus of your stomach, and then your stomach can actually be damaged by its own stomach acid. And that can give you ulcers. That's what Helicobacter pylori does. Okay. Chlamydia is a gram-negative bacteria that is an STI. It's actually the most common STI in the U.S. So it can be spread from partner to partner because it likes to live in the human urethra. And then spirochetes are technically gram-negative. So remember, spirochetes are the little bacteria that look like little worms because their flagella is actually embedded within the cell wall. And the two you need to know are Treponema and Borrelia. What's Treponema cause?
Syphilis.
Syphilis. So this guy causes syphilis. And what's Borrelia cause? Lyme disease. So this one's pretty common. So definitely know those two. Remember that spirochetes are technically in the gram-negative family. And let me see. And that's it for chapter 11. That's the big highlights. So any questions about that? No? Okay, let's take about a 15-20 minute break. Meet me back here in 15-20 minutes, and we'll just do lab in here.